Good afternoon. This is Dr. Dan Guerra from VerEvMed, recording from VerEvMed Studios in the Intermountain West of the American Northwest. This afternoon, we're going to do part 2 of a discussion we've been having about microglial reorganization of the central nervous system and how this relates to neurogenesis and synaptogenesis. As I mentioned in the first talk, in fact, even introduced, then in this second talk, we're going to go through another paper. I'm going to show you more detail about microglial activity in the brain. But we're also going to introduce and heavily embed this discussion in epigenetics. And then finally, we're going to try to develop a current theory that I'm generating about how the brain actually learns and develops via the immunoepigenetic phenomena that we've been describing and which is rampant in the literature. So let's get started and have some fun. All right. So first of all, let's get the slideshow going, right? Let's do that. Okay. Oop, wrong slide. This was part 1, you see. You get to see it in fast motion. Boom, boom, boom. See? And now we're here. Okay. All right. That's me. I'm Dr. Dan Guerra. That's my email address. This is my Facebook address down here. You can get to that Facebook any way you want to. And in that Facebook compendium, you're going to find all these VerEvMed lectures. You're also going to find some philosophy and lots of other scientific things. But remember that we are recording from VerEvMed Studios, and VerEvMed is our company. There is the VerEvMed email address. Contact us through that to become a client. And also that is the VerEvMed, of course, address to go directly to our website. Okay. I'm not going to talk much about VerEvMed today, but if you've been watching these, you know what we do. And these video presentations, these lectures are to exemplify the kinds of things we can do when we dive into the scientific literature as scientists. Remember that I am a biochemist, and so all of the things that I discuss is from that biochemical point of view. All right, so let's get started. Today we're going to cut through what I call the diaventome. Now, if you look that up in the dictionary, you're not going to find it. You're not going to find it because I invented that term. Okay? So the reason I did is I had to come up with something new to describe a mechanism that I believe is central, foundational, and coherent with what we see in the development and maintenance of the central nervous system, and therefore vis-a-vis the human brain, and how the human brain might actually then interact to generate what we call the human mind. I know that's a lot to think about, but that's really where I want to go with this. I want to generate ultimately a theory on how brain and mind function together. And that means you have to do a lot of cellular physiology and a lot of biochemistry, and also look at a lot of clinical associations and permutations that can help us understand this, and maybe even animal models. All right, so this is cutting through. So this actually was done this week. This is my pickup truck, and as many of you know, I cut wood to heat our house here in the Northwest, and this is indeed what I was doing here. This is a white fir tree that I cut down, and now it's all blocked and cut up and put in the back of my truck. I did that just a couple of days ago. You can see that up here in the Clearwater National Forest / St. Joe National Forest, we still have snow. And this is actually April. Today is April 13th. This was all taken just a few days ago, on April 11th. All right, so let's get started. All right, this comes from a paper in the journal Immunity. You can see that it's recently published just last month. In fact, just a couple of weeks ago. So let's go through some of this paradigmatic introduction. Microglia, as everyone knows, are the resident tissue macrophages of the brain. They're crucially involved in scavenging of dying cells, pathogens, and molecules through PAMPs. Now, PAMPs are pathogen-associated molecular pattern receptors. This is how things function. You have a receptor that recognizes, let's say, a foreign antigen that then induces something like an immune cell, like a microglia, and then once that microglial cell is activated, you then get the potentiation of an immune response. It's basically how things function. Now, the dysregulation of microglia can result in neurodegenerative and neuroinflammatory diseases. And that is, of course, got its own name. Those are called microgliopathies. Like that term, right? Now, it involves microglial colony stimulating factor 1 receptor, that's CSF1R. You find this in hereditary diffuse leukoencephalopathy with spheroids. That's a disease that can be diagnosed for those who are unfortunate. That's called HDLS for short. That's the acronym. Also, you have the ubiquitin-specific peptidase, or USP18, and the CD33 in Alzheimer's disease, and also TREM2, another potential mutation in that gene, in frontal temporal dementia. Okay, so these are all microglial-associated neurodegenerative diseases of the brain. Together with macrophages at CNS interfaces, such as the meninges and the perivascular spaces, microglia are derived from erythromyeloid precursors, or EMP, from early prenatal sources exhibiting no turnover, typically, and a low random self-renewal rate coupled to slow homeostatic apoptosis. That means final turnover, death. That's how microglia normally live in the CNS. Macrophage activation requires, microglia are a type of macrophage, as you know, requires precise regulation of gene expression that is governed by epigenetic mechanisms such as DNA methylation and chromatin and histone modifications, covalent modifications usually, but there's also a lot of hydrophobic interaction there with the membrane. So we've discussed all this in previous talks, so I'm not going to go through all the foundations on this. Now, histone deacetylases, or Hdecks, are histone-modifying enzymes that, along with histone acetyltransferases, or HATs, reversibly regulate the acetylation status of histones. So if you acetylate the histone protein, which is cohering with chromatin, you're going to modify the expression of those genes in that chromatin. Okay, that's all nucleosomal, right? Okay, so you get the idea that the turnover of acetate, so the more acetate you pump into the histones, the more you open up the chromatin, the more you get a global level of gene expression. So an open DNA or open chromatin, more gene expression, more transcription. A closed or condensed heterochromatin, less gene expression. But there's differential regulation of those genes. So Hdeck might shut down the global level, high amount of high copy number messenger RNAs from multi-gene families, sure. But it doesn't mean you're going to get a reduction in other gene expression. In fact, you might get an amplification thereof. So it's a differential regulation the Hdecks and the HATs present to chromatin. And these are all epigenetic phenomenon, right? That is, they're covalent modifications of DNA and cohering histones in the chromatin that relate to gene expression, but are not directly sequential. That is, they're not about the DNA sequence itself. Therefore, mutations can or cannot have an effect on the epigenome. So we have lots of Hdecks, lots of isoforms of that enzyme. Here we're talking about 1, 2, 3, and 8. And those 4 Hdecks can assemble themselves into a multi-component, multi-subunit co-repressor complex. And they often act basically, when they do that reorganization within the membrane, which requires certain membrane lipids, which we talked about in the past, such things as gangliosides and certain types of other sphingolipid species, as well as glycerol-linked lipids, as well as cholesterol. Anyways, that reassembly of those Hdeck proteins in the membrane, say in the microglia, will affect ultimately a phenomenon known as transcriptional repression, as I said. Right? So the more acetate you remove, you're deacetylating, right? You're removing acetate, you're collapsing the DNA, the chromatin, and you're reducing overall transcription. So it's called transcriptional repression. Sure. All right. So let's do some, okay, so that's an introduction. So let's do some hypothetical deductions, and let's summarize some of the event ontology. Sorry for that misspelling. So Hdecks should be studied to improve our understanding of microglial mechanisms that may control the CNS process. Asking question like, what is the role of Hdeck1 and Hdeck2 in the maintenance of microglial identity and function? Okay, that's the kind of question you would ask. Of course, you ask this every day, right? When you're buying a cup of coffee. Now, assume that active Hdecks should minimize microglial, that's MG, gene expression to some threshold, as I've just been saying the last 5 minutes. Assume also that a certain threshold should be tolerated, but beyond that, there's an impairment of microglial function, which could lead to maybe central nervous system corruption and maybe even degeneration of the CNS, or maybe even oncogenesis, right? Such as glioblastoma. Okay. Now, implemented, constitutive, and inducible chemokine CX3C receptor 1 promoter-driven Cre recombinase combined with conditional alleles for both Hdex 1 and 2 were used to generate mice, this is the study we're looking at here, whose microglia lack those two enzymes, lack those Hdex, from development in normal adulthood and or during neurodegeneration. So that's what they've done here. So basically you have a conditional allele. Now how's that work? Well, Cre recombinases work as a homologous recombinase. Homologous recombinase is what they do is they recognize certain flanking regions, and the flanking regions will flank certain genes, and when those flanked regions of those genes are recognized in trans by the Cre recombinase, those portions of that gene are removed. So you can use Cre to remove intervening sequences and thus activate the expression of certain genes. Or, of course, the other way, you can ablate that. Now, when you use this chemokine receptor promoter-driven Cre recombinase, basically then you're allowing chemokines to generate this recombinatorial conditional expression of Hdex. And in this case, what we're doing is we're shutting down the expression. Okay, so this is using homologous recombination to control gene expression, something we've been doing in whole animals, okay, the so-called knockout mice for now 20 years. So this is not new technology by any means. Cre recombinase is a standard method for doing this. Okay. So now, bracketing those initial assumptions of what we might be asking those questions about and what we're assuming is right, I'm going to tell you, like right now, what this paper is going to demonstrate, because I want you to have this running through your mind as we go through it. Not to try to control how you observe the data or how I interpret it, but so that you kind of know where we're trying to get to. And this paper does get here. So if you have that in mind, that telos in mind, might help you to follow through with what we're talking about when we get into the molecularity of the data. Okay, 3 things. Prenatal ablation of HDAC1 and HDAC2 caused, according to this paper, a spontaneous microglia impairment, including blockage or of proliferation and enhanced apoptosis. Okay, so you don't get a proliferation of the microglia, and you actually induce apoptosis. So that means you're taking the microglia out of the equation, okay, by removing the activity of the Hdeck. Now the Hdecks remove acetate. So if you remove the activity of the Hdeck, what are you doing? Okay, if you're not deacetylating, okay, so if you remove the deacetylase, what happens? You have more acetylation, right? Okay. Now, postnatal deletion was largely compatible with microglial viability and function. Isn't that interesting? Okay. HDAC and HDAC1 and HDAC2 were able to prevent microglial exhaustion and dysfunction in a mouse model of Alzheimer's disease, leading to a reduced amyloid load and improved cognitive functions. That's basically what they're going to show here. Okay, so they're going to show that removing those activities actually is going to prevent microglial exhaustion in the end, okay, because of its effect on proliferation and apoptosis, part 1 there. In part 2, it's going to be compatible with microglial viability and function. So in other words, the microglia are going to be working okay, but in the end, it looks like it's going to repair the mouse model for Alzheimer's disease. And the way the readout is by looking at a reduction in amyloid load going to be the A beta protein we're going to be looking at. Again, refer to previous talks to know about A beta's relationship to AD. And in fact, in this model that we're looking at, you can also show improved cognitive function in this mirroring model. There you go. That's what we're going to be going for. So now let's look at just the Hdeck expression profiles in embryonic T mature microglia. Okay, so what we're looking for here is we're going to be looking at the level of expression of microglia. On the left are heat maps. Remember the red color shows more expression. The fainter red into the pink and then the blue colors show less expression. Okay, on the log score. And on the Z-score, you get the blue coloration, which gives you a differential level of understanding the expression of the genes that have been modified by using the Krieger combinase. To the right there is showing you the actual data, the relative expression of the Hdecks 1 and 2. And what you're seeing here is the embryonic cells. Okay, those are early stage of microglial cells. Okay, and then as you proceed through the adult, going to the adult stages. So A1, A2, the actually embryonic MGs, these are still getting to MG. Then you have embryonic microglia. Then you have adult. And what we're seeing is both Hdeck1 decreases in activity as you age the microglia. Hdeck2 decreases as you do that. And here's the actual relative expression using this Z-score. So either using the log score or the Z-score, you get rid of Hdeck expression. This is just the first thing you do when you do Krieger combinase research. Did you decrease the amount of Hdeck? Looks like you did. So therefore, anything that follows, you can argue, is associated with Hdeck expression. Okay. There's a caveat to that, as you probably know, but follow with me. That's what we do when we do knockouts. Now, more heat maps, lots of cool data here. Wow, look at this stuff, right? This is just looking at what Hdeck expression does to the family of genes that are controlled by the acetylome. Okay, so all of this, just part one up here, all of these are genes, okay, that are controlled by Hdeck1, and there's Hdeck. Okay, so what we're telling you is that all of these genes are normally controlled via the expression of Hdeck. Okay, so the gene interaction with Hdeck expression in either erythromyeloid precursors or embryonic microglia, that's down below. So this is early, this is just the erythromyeloid precursor to microglia. You see a lot of genes are controlled by Hdeck1 and some by Hdeck2. Okay, it's what you're seeing here. Now look at how this works. The red, okay, I know you can't see all this, you don't need to, but the red is up-regulated, okay? The blue is down-regulated. So you get a differential control of expression over acetylation patterns, right? And these are down here with more confidence, predicted activation and predicted inhibitions. This is showing you the different nuances of color to generate these expression trees. Using the data from the heat map, which is looking at expression of all these genes, is to all gene nomenclature here along this bottom x-axis there and there, okay? So you got erythromyeloid precursors in E and H, okay? So that's here. And then in these down at the bottom, you've got embryonic microglial cells. So what you notice, okay, by looking at this data, what I notice and what the authors point out is that Hdeck1 and Hdeck2 mediate gene regulation, both in terms of number of genes and their relative expression level, were maximal in microglia prior to birth and were reduced as you get to older or mature microglia. So the number of kinds of genes that get controlled by acetylation, because they're comparing the Hdeck activity, right? Hdeck1 and Hdeck2. Hdeck's up here, 1, Hdeck2 is down here, right? So there's a differential between those two deacetylase proteins in terms of which genes are being regulated. There's a lot of overlap, of course. But what you're seeing is that there's a difference, okay, between when you go from the early stage of microglial development to the later stage, more mature microglia, where we actually are in a microglial phase. So less genes are controlled. That's really important to know. This is basically your background or your foundational level of gene expression as related to Hdeck. That's what we're talking about here. Again, this is foundational to understand when you're going to knock out this gene, which is what you're going to do with the Cre recombinase, manipulating what? Remember what we're manipulating, that chemokine promoter, okay? Chemokine promoter is a constitutive promoter in these microglial cells. So you use that promoter to control Cre expression. Cre expression controls Hdeck expression. That's going to vis-a-vis control at some relative frequency the expression of your genes you see here, either in the early stages of erythromyeloid precursor or in the later stages when you're looking at the embryonic microglia themselves. Okay, that's what we're talking about here. Okay, so this data is going to show us that prenatal Hdeck1 and Hdeck2 ablation via the Cre recombinase activation on the chemokine promoter induces microglial malformation. Okay, so here we're looking at how this is done. There's a little mouse. There's the embryonic stage, and this is the post-embryonic stage. Okay, there's week 2, 6 weeks, 16 weeks out. And we're analyzing during all those times. That's what these x-axes are, okay? And we're seeing the mouse, and we're doing this Cre recombinase, and that's the way you put the genetic markers in, showing that you're going to alter, you're going to flank the Hdeck1, Hdeck2 with the Cre recombinase, utilizing, you see here, the chemokine receptor. See, that's what that means. That's what that construct is. Let's take a look. Here's relative expression, always on the y-axis here. And down here, I will mention in a minute what these IBA plus 1 cells are. What you see is when you turn on the Cre recombinase, what you're getting is a decrease in the relative expression of Hdeck1, okay, as time goes on, the relative expression of Hdeck2 as time goes on. Now, the number of IBA1 cells, that's actually a marker for active microglia. And what you see here is that as you cause the Cre recombinase to be activated, you get a decrease in the overall active microglia. Marker for microglia, active microglia, is that gene expression. Okay, but when you move out further, you get more mature, you see, because you're altering all those gene expressions that you saw on the previous slide. The effect of Cre recombinase on gene expression basically is insignificant, except it still looks like it might be at 16 weeks. So there might be some significance here, but here it says no significant difference between these two, although remember, notice this has a pretty large standard error bar. All right, so what are these things reading out to look at? Let's look at dendrite length. So the amount of dendrite length, these are from the neurons now. Now we're looking at what's going on in the neuron. You're getting less of it with the Cre activity at the very beginning, the embryonic stage, as you're moving through to older and older microglia, and you turn on the Cre recombinase, less dendrite length. The number of branch points you're getting to decrease in that. So see how microglia is controlling this. The number of segments that are regulated by the microglia themselves, and the number of terminal points is all the ways of describing, as you saw in the previous talk, how neurons are being regulated by microglia. And you see that there's a decrease in all this. That doesn't mean it's negative. It just means that there's a potent effect of Hdeck expression on how neurodevelopment and how microglia form. Okay. So let's back up here a second and let's look at, so all this data here, all this activities, these are all from microglia. This is all looking at microglia, not from the neuron, but from the microglia. Okay. And it's showing there's some kind of microglial malformation. Remember that microglia can do good things, but when inappropriately activated, it can do bad things, such as neurodegeneration. Now let's back up and take a look at a paper published a couple years back in Science Reporter 2016. There's your reference. Now, what is this paper telling us? Resting microglia positively correlate with brain biochemistry by removing cellular debris, secreting neurotrophic factors, those are like growth factors, and recruiting immune cells for maintaining the CNS. Those are resting microglia. They're still active, but they're not activated. However, when you activate the microglia because of, let's say, some kind of incursion by a pathogen or some other stress phenomena, when you activate the microglia, it adversely affects brain biochemistry via the generation of reactive oxygen, nitric oxide, interleukin-1 beta, which is a pro-inflammatory cytokine, and TNF-alpha, which is also a pro-inflammatory cytokine. Those are all associated with neurodegeneration in the CNS. Brain cystine, okay, is essential during CNS development to avoid cognitive dysfunction. Now, that is a very complicated thing. I'm just bringing it up here because it's in this paper. And you can get a reduction in cystine in immature neurons via microglial uptake for GSH, because microglia need what? They need glutathione to do what? To remove the reactive oxygen. So they start taking cystine away from whatever cystine is necessary for the neuron. So that's one way of decreasing the amount of cystine that gets into the neuron, is by activating the microglia, which need more GSH, which reduces glutathione. It can also be affected, lower cystine uptake by the neuron, by inhibiting the cystine translocator, which is what XCT is. And that can also be induced by high extracellular glutamate, which happens during stress. Okay? So both of those things are going to cause a potential cognitive dysfunction. Too much glutamate, corrupting the transporter for cystine uptake, and also for an enhanced requirement for glutathione because of an activation of microglia because of an inflammatory response generating reactive oxygen. So, two more things you need to know before looking at the rest of the data. The reason I'm showing you this is so that you get this information so the data is understandable to you. It's comprehensible. Okay, first of all, what does tunnel mean? Tunnel means terminal deoxynucleotidyltransferase deoxy-UTP-NIC-N labeling. It's just a technique. Don't worry about it. It's not very complicated, really. So it's a method for detecting DNA fragmentation by labeling the 3' hydroxyl termini of double-stranded DNA breaks during apoptosis. So it's a marker for apoptosis. Okay? That's what it is, tunnel. Another thing you need to know is something called, we saw it a few slides ago, if you remember when I said about activated microglia, is the ionized calcium binding adapter molecule 1, or IBA1. That is a microglial macrophage-specific calcium binding protein with actin bundling. Remember actin? Actin works on making those processes, remember from the previous talk? And participating in membrane ruffling and phagocytosis in activated microglia. Okay, so this is a marker of those activated microglia that might be involved in ultimately phagocytosis because of its alteration of actin bundling, which is then participates in overall hydrophobic interactions in the membrane, all at the microglial level. So you're going to need to know all that just to understand this slide. Okay. So what we're looking at, another heat map over here, all these different genes, these are all the different genes and how they relate to one another. That's what this map over here is, how these genes interact. Okay. That's what the Z-score is going to provide for you. Absence of Hdeck1 and Hdeck2 during development alters microglial histone acetylation and transcriptome profile. So basically, we're just looking at what's going on with the acetylation and acetylation controlled transcription of all these genes. So just taking a composite set of data from all of that heat map data. These are genes involved in apoptosis and signaling. These are genes involved in cell cycle. These are genes involved in interaction signaling. These are genes involved in retraction of cellular protrusions, which relate to microglial activity of synapses. And this is collapse of the growth cone, which also relates to how microglia affect neural synaptic development or synaptogenesis. Again, from the previous talk. So you can see here that after you've induced Cree, these are all Cree data, you see a decline in cell morphology, cell growth and proliferation, cell death and survival. You get a decrease in that as well. Cellular compromise decreases by 6 weeks, and cell cycle changes in a bimodal fashion. When you alter the expression of these specific genes, you get this readout in terms of cell biology. So you're looking here, if you looked at this electron micrograph, this enhanced micrograph, you can see here how the microglia are affecting the neural development, okay? And I'm not going to blow that up for you, but that's basically what it's showing you with Cre+. You're getting a change in the morphology of these cells. And this is data showing you that when you turn on Cre, you get more tunnel. Remember that DNA fragmentation suggesting apoptosis. And you're also getting more activation of the microglia. Remember I told you that when you activate the microglia, you're also differentiating them terminally into turnover, right? So the more rapidly you push them in that direction, the resident microglial cells will go through turnover, but they're also being activated, thus acting to alter neural development. So you have to understand what's happening to the microglia to understand what's happening to the neurons. When the microglia are activated, they're going to go through a biphasic activity associated with the neurons. So hitting that just at the right place can alter how the neurons then are being altered by such things as the relative causative relationship of A beta protein to plaque formation. Okay. Now, what this data says is the absence of Hdeck. You see here we're getting lighter colors here in a lot of the expression of these genes, right, such as apoptosis. Absence of Hdeck 1 and 2, okay, during early prenatal microgliogenesis changes the acetylation pattern of the gene and also the expression, which is what you'd guess, okay? And so you get a acetylation pattern of defined promoter regions that regulate microglial survival and expansion, especially by modulating cell death, activation, morphology, and cell cycle. So all these things are affected by altering the acetylation pattern of all these genes, all in the microglia. And then that ultimately can affect what's going on with the neuron, which is going to relate to the activation, the turnover of the microglia. So you have to understand this is all microglia data, and ultimately it's going to impact on neural, that is neuron, biochemistry and cell physiology and cell degeneration potentially. All right. What else the data shows, I'm not showing you a big hunk of the data here, but summarize. Hdeck 1 and 2 are redundant. That means they overlap in their activity for steady state maintenance of adult microglia. If you knock out one or the other, you can still maintain some activity, some semblance of regular normal physiology. So there's a redundancy between the two isoforms of the enzyme. Microglia from Cre plus mice, sorry, I got to take these off, showed significant downregulation of Hdeck 1 and Hdeck 2 compared to control or Cre minus, but didn't develop any overt neurological phenotype for at least 6 months after induction. Quite a while. Genetic ablation of Hdecks from adult microglia reduced amyloid plaque and rescued spatial learning and memory deficits. Wow. So you see here, when you get rid of the deacetylase, remember, getting rid of the deacetylase means you're removing the enzyme activity which deacetylates. If you remove a deacetylase, what do you get? You get a more acetate in the chromatin on the histones, which means you're getting more global or overall messenger RNA transcription. Yeah. Which means you're basically slowing down, and this is how I see it, you're slowing down the lifespan, the half-life of the microglia from being non-activated to activated to being apoptosis to dead. You're slowing it down. Okay. All right, let's take a look at this data now. Again, here's your little mouse that you know that the mouse is controlled by the flanking of the Cre recombinase sites around these two genes. This is showing you again when they do it. This is 24 to 28 weeks out. This is when the analysis is, okay? You're looking at Hdeck 1 and Hdeck 2 activity. And basically, this is when you're looking now at the Alzheimer's disease model. There's wild-type relative expression of Hdeck 1 and 2. See, boom, the black here. Then you've got this FAD. This is familial Alzheimer's disease. Okay, this is I call it a 5X because it's back crossed 5 times in these mice. So this is a very strong AD penetrant line with familial AD now. Okay, this is different than the idiotypic one, but this is the one that uses a model for some studies in mice. You see that the relative expression of Hdeck is less, although not significantly so, between the wild type in the AD model, but much less in the knockout, the double knockout, the DKO, knocking out both Hdeck1 and Hdeck2 here. And okay, so this is showing you the relative expression, looking at either Hdeck1 or Hdeck2 separately and with the double knockout. So once again, you see the relative expression, okay, relative expression, and you're looking at a reduction in Hdeck activity. Okay, a reduction in Hdeck activity compared to the Alzheimer's disease model, and which is significant even to that, and certainly significant to the wild type, both of the isoforms of the enzyme. Okay, so this data here, I know there's a lot of it, but it's the end of the data. Basically, on these x-axis, y-axis, you're going to have all kinds of different things. Number of plaques formed. Let's look at this one in the cortex. You turn on that Cre recombinase. Remember, the red's always going to be the Cre recombinase, which means you're reducing the Hdeck. Less plaque in the cortex. This is the amount of soluble A beta 42 protein, which you know is the precursor to the oligomers, which is associated with Alzheimer's disease, even better than plaque is. Big reduction at 24 to 28 weeks, and even out there at 48 weeks when you turn on the Cre recombinase to ablate Hdeck. In the hippocampus, same thing, right? You get a decrease in the relative expression of that. In the cerebellum, same thing, okay? So all the way across the board, these are all good phenotypes telling you plaque and soluble A beta 42 are reduced compared to familial AD when Hdeck is KO'd. That means it's knocked out. Okay, that's just continuing to show you the same thing here. You know, there's cortex, there's hippocampus, number of plaques again. Here's more cortical data, the A beta protein, and on and on. Each one of these is insoluble A beta. Okay, you get a decrease of it out here at 48 weeks. The insoluble is bad player. You get less of it when you knock out the Hdeck, and that happens in the cortex, in the hippocampus. And this is now looking at the insoluble A beta in terms of how much protein is there. Okay, same thing. This is all good. This is what you want to see. Down here are some of the behavioral patterns. Percent time to target quadrant, that is whether or not you get a behavioral output that looks like a loss of cognition in this model of Alzheimer's disease. And what you're seeing here is the Alzheimer's disease, the mice. You have a reduction in this parameter, but when you ablate the Hdeck, you get back up to wild type. In fact, a little bit better. And you see this in these three different behavioral patterns. Okay, recovery, percent alteration. These are all readouts, behavioral readouts. And what this tells you is that Alzheimer's disease behavior disappears. Okay, goes back up. But here, this is actually what you want. That activity shouldn't be high. It's high in Alzheimer's, so it's low again when you do the double knockout. So Alzheimer's disease behavior disappears after Hdeck knockout. So again, this accumulated data is really amazing. It's showing you that controlling microglial epigenetically mediated gene expression will alter the overall microglial half-life in such a way that it will mediate the final neural output for Alzheimer's disease, bringing a familial AD population, to look more and more like a wild type. So decreasing plaque, decreasing the A beta, decreasing the insoluble A beta, and when you're looking at the behavioral output, it looks more like wild type than it did in the familial Alzheimer's disease model. That suggests that if you decrease those two Hdeck isoforms, knock them out, you may be able to have a potent, maybe pharmaceutical drug, okay? Knock out those two Hdeck to be able to be involved in maybe even late onset Alzheimer's disease. Because remember, this is way out there in terms of weeks into these mice. This is like adult and aging mice here. Let's summarize. Acetylation modifies non-stochastic chromatin, that just means non-random, that results in neurogenesis, neurohomeostasis, and neurodegeneration via microglial epigenetic reprogramming. So that links then the immune system to the epigenome. Important part of this paper. Okay. There's a control over the central nervous system microglial histone deacetylation. Okay, shown by this paper by using the Cree recombinase to cause the ablation in the microglia. So that control over CNS microglial histone deacetylation may be an important target in Alzheimer's disease and maybe even in other neuropsychiatric diseases. That's the hope for this kind of research. All right, let's do a dialectic. Okay, from a summary to the dialectic. Dia means across, and lectic means like a discussion thereof. The genome contributes to heritable phenotype of the CNS. We know that. So the genome's involved. Absolutely. The environment contributes to the heritable and non-heritable phenotype of the central nervous system. That's also well documented. That's your epigenetic effect. There's also stress phenomenon, xenobiotics, right? Drugs like tetrahydrocannabinol or ethanol or cocaine or heroin, things we talked about in 4 or 5 previous talks, right? All of that is part of the environment. Also, aging itself, because of the vagaries of the increase of reactive oxygen species, can be called environmental effects, as well as developmental sequences leading to senescence. So that's important. Now, so you have genome, you have environment. Then look, wow. This is the really important player here that we just have been looking at. And this is backed up, by the way, from numerous papers in the literature. This isn't just as few papers we've looked at. There are hundreds of papers just in the last couple of years that are looking at the same thing, not just in microglial-mediated neural interactions, both pathological and healthy, but in T cells and B cells, and lots of other immune system-associated things, such as even epithelial cytokine and chemokine metabolism. So an epigenomic transcendency, that means sitting on top of, over gene expression, acquires and early development and then outward to maturation, and extends neuroplasticity. Remember we talked about what neuroplasticity is, right? That means you can modify the synapse in such a way by carrying out differential synaptogenesis and trogocytosis, or alteration of the membrane in the presynaptic cleft, thus altering the amount of neural transmission that occurs across that synapse, thus regulating how genes affect the action potentials that relate to how ultimately neural signaling leads to things like the movement of thought patterns in the brain. Okay? That's all about the endophenotype. That's all biological psychiatry, right? Or neurochemistry or neurobiology. So neuroplasticity, which is usually considered good, but if you overextend neuroplasticity, that can also cause a fuzziness of those synapses, which causes things like confusion, right? Or even worse than that, can actually cause degeneration, or in some instances, even oncogenesis. There's also elasticity. And that's something you don't see in the literature. That term isn't used, but it's all over if you look at lipid metabolism. So when a plastic event occurs, that means you make an impression on something, and when you remove that effector molecule, that impression stays, okay? That's what we normally think about when we're affecting the neural circuitry, right? You learn something, you've altered synaptogenesis and synaptic alterations, you've altered neural transmission in such a way that you maybe even can now generate coveted memories, such as when you had a wonderful picnic lunch with your girlfriend when you were 25 years old, okay? Where's that memory at? That memory is by all of the different neurons and all the synapses associated with those neurons that were tailored during that time via microglial stimulation, that are now firing and wired together in such a way that when you try to recall that event, it comes back into your brain and it looks like a movie. Okay. And all the other things you've learned, not just things like that, like a beautiful day up in the mountains. Also, when you learn what a microglia is, when you first learn what glycolysis was, or all importantly, when you learn all those different metabolic pathways leading to and leading away from lipogenesis. Those are the really important ones, right? Okay, now elasticity is also important. Elasticity means you have an impression, but then when you remove the stimulation, the impression goes away. So like a rubber band's elastic, you pull it, and when you quit stretching it, it moves back, it falls back into place. So elasticity is also important. So that's like short-term memory, or that's like the kind of things you don't need to remember long-term, or the kind of neural circuitry that, if it were maintained, could cause that fuzziness or an alteration in the smooth running of the neural system, therefore decreasing things like cognition and emotion. So you need elasticity too. So epigenomic transcendency over gene expression acquires and extends neuroplasticity and elasticity that results in disease, health, and eventually the processes that control cellular mortality and lifespan. Okay? Because all of this is eventually coming to an end at senescence, when the organism dies, when the brain dies. Now, I want to remind you, like we did on the first part of this talk, about the immune system. You have two major lineages, the myeloid and the lymphoid. Myeloid gives you the granulocytic and the monocytic cell lines. The granulocytic are the neutrophils, basophils, and eosinophils. These are all part of the innate immune response. Macrophages, Kupffer cells, dendritic cells, all really important as antigen-presenting cells, for example, also for digestive phagolysosomal digestion of antigens, etc. The lymphoid cell lineages, I think most of them know about natural killer cells. They go out there, they find something that's bad because of the ornamentation on the surface of a cell. It goes in and kills the cell outright, no questions asked. Or B cells, which of course are more nuanced. They make the immunoglobulins, which are working with complement, and also with T cells, can affect the destruction of foreign antigens or organisms like viruses or bacteria. And then the T cells, which are the coolest of all, because they're involved in basically the control of all the immune system. And you have T helper cells, T suppressor cells, and cytotoxic T cells, right? And so these all have gotten kind of newer names, right? So the suppressor cells are now called Tregs, for example, Treg cells. But this is just from an immunology textbook that I used a few years back when I was teaching immunology. But you get the idea. Lots of different cells. Now, why am I telling you this now? Because we were just looking at, wow, what are we looking at here? Macrophages, and even one subtype of macrophage, the microglia. Guess what? Epigenetics is involved in all of these cell lineages. So if you want to find a model system to be able to control neural development, you need look no further than the immune system. The immune system is in place. It's functioning from the first days in utero, when the embryo is first formed during utero, before embryogenesis, before fetal development, throughout the parturition process, post-parturition, the first 2 years of development, through puberty, post-puberty at age 20, where I told you where the brain starts functioning, where it's actually sensible, and then all the way to when you get older and older, and then you start becoming maybe a little bit insensible when you start getting older, right? That's happening all the time. The immune system is functioning all the time. And so it is there to generate a repertoire of activity via epigenetic programming and reprogramming by being stimulated and affected, and also by being suppressed by environmental cues, which gives you all the learning that it goes on, plus all the vagaries that go on by living in a world. Okay, so think about that. Now I'm going to do a little bit of I'm going to get into this theory, and I can see that I'm kind of overdone a little bit with time, so I'm going to just try to present this theory with you today, and then we're going to go talking more about it by using the scientific literature. Now, here we go. Before I can get to the theory, I want to explain a little bit that I didn't in the last session. I want to tell you why I'm really interested in trigonal planum. I wanted to find a model in chemistry, because chemistry is like geometry to a scientist. Chemistry is very exact, right? So if you've done things like solve for how you get angles in a geometric system, you know that if you have the right preconditions and you know how the laws work in mathematics, you can come to a conclusion. Well, chemistry sort of works that way, although of course it's always not 100% certain. But chemistry we know quite a bit about it, right? Because we invented it from nature. We invented chemistry from nature to help us describe nature. That's how it works. That's how science works. Just an invention after all. Okay, let's take a look at this. Valence shell electron pair repulsion concept, which is, if you have ever taken basic chemistry, you've heard of this. Case in point is the trigonal plane. Now, what is this? Okay, this is carbonate. Okay, carbon atom with 3 oxygen atoms. It's a double bond to one central oxygen here. This is looking at the electrons associated with the bonding and the electron pairs that are not involved in the bonding. Okay, so this VSEPR, which is valence shell electron pair repulsion concept, allows, I know you're saying, what is he doing? This is chemistry. Don't worry, you'll see in a moment. Chemistry is important. What do you mean, what is he doing? Of course I'm doing this. You have to do this. Allows that electron pairs will repel each other, whether they are bonded or in lone pairs because of electronegativity of the electrons, okay? You got too much electron dense region, it repels, right? That's what happens, okay? So the VSEPR will then result in molecular geometrical arrangements that's going to minimize the repulsion, okay? Always get to the minimizing of that because it has to do with energy equivalence. Molecules that contain a central atom, for example, this one here, a central atom like carbon, which I think we're all familiar with that because we're carbon-based, bonded to 3 oxygen atoms like here, okay, are then restricted when you look at this molecule to actually a 2-dimensional trigonal, which is simply fancy for triangular plane. Okay, so that's why it's 2 dimensions. That's why it's shown here. And this has a form of -2 charge, by the way. That's why you make bicarbonate from carbonate. Okay, very important actually in blood chemistry. A topic of one of my previous many lectures in teaching and also even in some of the VerEvMed lectures. Okay. Hemoglobin and all that. Now, such a molecule is carbonate, where one oxygen is double bonded to the central and the other oxygen atoms are single bonded. I want you to think about this when I showed you the next slide. That's why I'm showing you this. I'm trying to imprint on you a chemical, sort of a chemical and geometrical proof. Okay, they say, well, this is what we know about how bonding works. And here's a very simple molecule for sure, carbonate. But it's giving you all of a sudden all this information about, repulsion of electrons and how things interact and what bonds are, right? Just by looking at a very simple compound. Okay. Now, ask the question, why am I talking about chemical bonding? Okay. Why am I talking chemical bonding? You could ask that question. Here's why. Answering. Chemical bonds are not material substances. No, they're not. You can't go and look for a chemical bond and say, here, I'm going to weigh this chemical bond. It has this mass. Okay. You can talk about atoms having mass. Even that is a hypothetical deduction. Okay. Remember, chemistry was invented by us to describe nature. Okay. Chemistry isn't like there unless we put it there. That's how all of our knowledge works, because we all work with our senses or sensoria to explain the phenomenological world. Chemistry, which is just one of the natural sciences, is just doing that, only at a very nice, detailed level. All right. So chemical bonds aren't really material substances. They are representations of dynamic energy confluence, where energy is able to be confined, or there's a confluence of electron-dense regions where you get a bonding, such as a chemical bond that you just saw in the carbonate molecule, or in any molecule, DNA, RNA, protein, lipids. Lipids super important. Dynamic energy is obtained temporally as well. So that gives not only that you get this dynamic energy confluence just looking at the bonds in static mode on that plane. Remember, this is through time. You don't just have a carbonate molecule and it instantaneously is there and not there, right? This isn't ex nihilo synthesis. It comes from somewhere and it's going to go somewhere. Yes, of course. So dynamic energy is obtained temporally. That means it's a time signature, and therefore an event rather than a substance. So that allows me then to say that event ontology, sorry I misspelled that, I always do that, takes into account the dynamics of neural circuitry. So you see it in chemistry, you know you're going to see it in biochemistry, which is based on chemistry, and so you're going to see it in cell physiology, and cell physiology is the basis of what? Neurochemistry, and neurochemistry is the basis of biological psychiatry. Okay, so you're already all the way up to behavior and such things as neurodegeneration and such things that you find in the DSM-5. Right? All right, and also all healthy brains, which I know all of you have. Now, we're talking event ontologies here, not substance ontologies. That's because of chemical bonding. Get all the way down to foundations and coherence with these truths, small t. Neural circuitry then obtains via immunoepigenetic phenomenon. We already saw that. I just showed you a couple examples today. The previous talk that I did a couple days ago, same thing. And most of the talks I've done about neural chemistry, what we've done is talk about this neural circuitry and how it's modified by either epigenetics or the immune system, okay? Because it's all over the map. I told you microglia, big deal in the central nervous system, but we can also talk T cells, B cells, and all the possible ramifications of cytokines and chemokines, and all the other neurotrophic factors, all being linked to both an immune-based response, what they call immune, which is normally considered just defense, right? I'm saying that it's a bad term. We shouldn't be calling it when we're talking about what the immune system players are doing in the brain, or in fact in the whole body, when they're not directly dealing with the detection, the elimination or sequestration of invading organisms or stress systems or antigens, right? So immune system, when you keep on saying that, people keep on looking for, where's the virus? Where's the bacteria? Where's the pro-inflammatory response? You don't necessarily need all that, because these immune cells are functioning quite fine, thank you, like we saw in the resting microglia, right? They're functioning and they're considered immune cells, but they're doing something different. Same structure, different function, we should have different terms. The trigonal planar model I went through is the simplest representation for neural system dynamics, my argument here. Now, we could go into a more complex thing. We could do tetrahedral. We can talk about multidimensionality. I only gave you a two-dimensional grid. I'm already telling you times there, so really it's a pseudo-planar system that we're talking about here, which I mentioned in the last talk. But it's the simplest way I can represent it. And it's not because I think that we need to be simple. It's because using the simplest representation, we can then move from there to start looking at, first of all, how to design experiments. First of all, how to think about this, come up with hypotheses, then make some deductions from those hypotheses, and then using those hypothetical deductions to start generating what? Research questions. Those research questions turning into experiments. Those experiments, if they're designed well, turning in results. Results then finally being induced to learning more about nature. What nature? In this instance, neurology, right? The human mind, ultimately. All right. So here is the final, like my little synthesis of this. And I'm going to give that a new name for how the immune system plays. I'm going to call it a diaventome. Now, where do I get that? It's from the word dialectic, D-I-A, which simply means across or through. And then I've already told you why it is an event ontology, not a substance ontology. And I don't want to call it an immunome or something like that, because it's no longer an immune system when it's functioning at this level. So I'm going to go completely basic on you, and I'm going to use a term that could perhaps be called upon when we want to look at how epigenetics and the immune system come together to describe and to help us interpret better what's going on in neural biochemistry, neural cell physiology, ultimately how the brain works, the central nervous system works, and then maybe eventually understanding how the brain is interpreted into mind. Okay, here we go. Here is my plane, my trigonal plane. In the middle, you can consider this to be the brain itself or the self itself, right? Because the brain is where the self is found, right? So whoever you are, your self is embedded in your brain, which is translated into your mind, right? Because I want to go all the way down through and back out, all the way down and back out, so that we can see how this particular theory is trying to help us get at perhaps research questions that can help us further understand what it means to be a self and what it means to have, at least what it means to have a brain-mind continuum. Okay, so I call it diaventome. What are the components? What are the three things bound to the self or to the brain? It's a 7-pointed star. There's a reason I chose 7-pointed. I'll go about that. I'll discuss that at a later date. One is the genome. We talked about that. The other is the environment. We talked about that. And the third is the immunoepigenome, which I also just introduced. Now notice the genome is pink. The environment is green, right? You get that, the environment, right? And then the epigenome is supposed to be kind of like a fluid mosaic of the two. Now, of course, genome is pink because they do that for the geneticists, because I know some geneticists and I know they really like the color pink. Okay? Biochemists, we don't like that so much. We prefer the blues, but what can I say? All right, now take a look at what I'm trying to say here. Not trying to be funny, just trying to throw a little bit of humor into this because I know some of you are thinking, what is this guy talking about? Let's do this. Neural networks are processes becoming eventual in nature as opposed to substantial. They're not just substances. They're not just like this happened and that's it. There's not just taking something like the brain and saying it's just a hunk of membrane and it has neurons and neurons form synapses and it has glia and microglia and there it is, just this pasty wet substance, right? No, the brain is a process, right? It's a process. It's an event ontology. It becomes constantly, it's constantly changing. So it's becoming eventual. And that's what its true nature is. And that's different than being just something that is material, substantial. That's the biggest problem between looking at how the brain and the mind come together. If you think the brain is just a substance, matter, right? Material substance, right? Substance just means below or standing below what the matter is. That's what substance means, substantial means. So that's not going to work here, right? You need to have a dynamic process ontology or event ontology to understand how the brain functions through time. So I say an event ontology that reaches across, that's the dia, the neural network of genetic and environmental interaction, see, genetic and environmental interaction, is articulated and synthesized through, synthesized via a priori synthesis at the first level, epigenetic modifications of the classical immune system, thus creating a responsive, perpetually responsive, plastic and elastic, remember I told you the significance of that, you don't remember what you don't need to, memory field capable of learning, ideation, imagination, and even understanding through time. Thus, it's a process event ontology. The pattern of events appear as energetic fields. Okay, so that's the whole idea why I was showing you chemical bonding, because that's nothing more than energy fields, right? But they are dynamic. They're not just material substances. In fact, they aren't that. The pattern of events appear as energetic fields rather than particles of matter. That's my whole point about showing the chemical bonding, much like chemical bonds. So what am I saying? Central individual agentically, that means that making free choice of will, interacts through the events obtained across the inherited genome, okay, the facticity of what you inherit, the changing environment where the organism finds itself, the mind finds itself from the brain, right? And the transcendental working above those, right? Not something mystical or mystifying. The transcendental just means above the system. Immunoepigenome through time. So that's what I'm now calling, that's the definition of what I'm calling the diaeventome. And I'm arguing that this system is the best, simplest way we can perhaps now to understand and describe what the neural system is that makes up the brain and how the brain then translates to the mind, therefore avoiding Descartes' paradox. So here we go, more about diaeventology. It's an adaptational processive accounting. So all I'm doing here, this would be the study of, because it's an ology, right? An accounting. Now we got O-L-O-G-Y, comes from logos, means an accounting of, the diaeventome. An adaptational processive accounting of the rational human mind through cellular molecular event ontology. Because I'm a scientist, I'm not going to say that there's some voodoo between the brain and the mind. I'm trying to find the mechanism thereof. The mechanism thereof is that three-level interaction with the genome, with the environment, and then the immunoepigenetic mechanisms that occur constantly throughout life, right? From gestational all the way to death. The immunoepigenomic induction of neural, endocrine, and metabolic responses to the micro and macro environment, what happens inside, what happens outside, through classical constitutive surveillance and acquired effector, that's your immune system, constitutive surveillance, acquired effector, cellular and humoral, right? Cell-based level and liquid-based level, the blood, for example, the lymphatic system, defense stratagems, that's your immune system, okay? Using reversible covalent modification Okay, that was the acetylome, methylome, things like that, and hydrophobic interactions, which I'm going to develop in future talks, of nucleic acids, proteins, and lipids. This may become the central molecular theory. It could become for free will driven, because you need the agency of the system in order for this to be able to encounter the environment, right? So free will driven, agency based, existing individual, there's an existential throw in there, adaptation and knowledge acquiring epistemology that explains the core event ontology of human existence. I know that's a really broad sweeping, but that's what I like to do. I like to take my scientific side, my philosophical side, and other elements of what I work with here as my base structures and try to explain what I see in the scientific literature. And we're going into this theory. So that's what I'm doing here today. All right. So I'm going to stop there, let you digest all of that. It's now the weekend. This happens to be Friday the 13th. Favorite type of Fridays are Friday the 13th for me. I don't know why. I always have good luck on these days. Showing you a diametrical opposition maybe to how other people look at it, but not me. Love Fridays the 13th. Always good things happen. At any rate, let's finish now. And hopefully you found it, if nothing else, entertaining, but also maybe hopefully you learned a little bit, at least about how microglia are activated, and maybe a link to Alzheimer's disease. If you don't want to buy anything about my theory, that's fine. But there it is. I put it out there. Okay, that's me, Dr. Dan Guerra. There's my Facebook page where this is going to be posted soon. We are VerEvMed, where we are scientists verifying published evidence in medical biosciences. That's actually what we do for a living. There's the email address for the website, and there's the website itself. There's my email address if you want to contact me directly. I try to respond within 24 hours. Again, what is this? This is, of course, the Continental Divide in lovely Colorado. Thank you very much for your attention. And I know some of you have been waiting for this to be over, and others of you maybe are sad that it's over, but whatever, some melancholy always mixed with sweetness, right? So let's just say what I often say, oh, look, just a little over an hour. I think I did an okay job. What I always like to say at the end of these lectures, and very meaningfully, best to you, and bye for now.